Hi, welcome to my YouTube channel. This is the part 2 of memory based ASRB NET Prelims and Senior Technical Officers exams question conducted on the subject plan Pathology during 2021. Let us start with the first question. Gene that is responsible for the protection of tDNA from nuclease in Agrobacterium tumefaciens is, out of the four options, the correct answer is A, that is VIR-E. So now let us try to understand what is the meaning of other options. So VIR-C, in Agrobacterium tumefaciens, the VIR-C gene plays a crucial role in transfer of tDNA from the bacterium into the plant host during the process of crown gall disease development. And the VIR gene is one of the genes within the VIR region of Agrobacterium tumefaciens, that is TI, that is tumor inducing plasmid. The VIR region contains a set of genes responsible for transfer of tDNA and other virulence function required for successful infection of the plant. It is interesting that the specific role of VIR-C gene is to encode a protein called VIR-C1, which is involved in the recognition and processing of the tDNA border sequences. These border sequences are very much essential for accession and transfer of the tDNA from the TI plasmid to the plant host cell. Besides that, the VIR-C gene and its encoded protein, that is VIR-C1, are crucial component of the Agrobacterium tumefaciens infection process. It enables the bacterium to transfer the tDNA into the plant host and cause crown gall disease. Followed by, that is VIR-G. The specific role of VIR-G gene is to encode a transcriptional activator protein called VIR-G. This is also known as VIR-A. The VIR-G protein functions as a sensor of a specific plant-derived molecule known as phenolic compounds or phenolic inducers. These phenolic compounds are released from wounded plant cell and act as signal for Agrobacterium tumefaciens to initiate the process of tDNA transfer and infection. The VIR-G gene and its encoded protein, that is VIR-G, plays a central role in the regulating the expression of the VIR genes in response to the presence of phenolic compound from the wounded plant tissues. This activation of the VIR gene is a crucial step for Agrobacterium tumefaciens to initiate the infection process, transfer tDNA into the plant cell, as well as cause crown gall diseases. Now let us see the last option, that is VIR-A. VIR-A, its main role is to encode a protein called VIR-A, which functions as a membrane-bound receptor or sensor. It is localized in the bacterial inner membrane, and its structure includes both an N-terminal sensor domain facing the periplasm and a C-terminal cytoplasmic domain. So I think you know what is the periplasm. Periplasm is the space between the inner as well as outer bacterial membrane. Next question is, bacterial fruit blotch of cucurbits is caused by, the correct answer is Acidovorax avini, subspecies citrulli. So before answering this question, we should understand the symptoms of bacterial fruit blotch of cucurbit. They produces number of different symptoms like angular leaf lesions, stem lesions, fruit lesions, fruit cracking, and reduced fruit quality. So these are the main symptoms produced due to this disease. Besides that, they also produces some secondary infections as well as spread the disease with the help of rain splashes onto the plant during the rainfall or the irrigation. And this disease can also be spread through infected seed. Next question is, the first sequence bacteria is Haemophilus influenza, and the first sequence plant pathogenic bacteria is Xylella fastidiosa. So Haemophilus influenza, which is responsible for causing the respiratory infection to the human being. On the other hand, Xylella fastidiosa, the first sequence plant pathogenic bacteria, is a gram-negative bacterium that causes various plant diseases including Pierce disease in grapevines, citrus variegated chlorosis, and almond leaf scars. Next question is on killer protein produced by double-stranded RNA is found in. There are 4 options. Number 1 is Magnapordia oryzae, followed by Rhizoctonia solani, and followed by Astilego maris. And the correct answer is Astilego maris, that causes smart of maize. Number of oospores produced per oogonium in oomycetes is, the correct answer is 1. RxLR is a type of effector or secreted by, the correct answer is Phytophthora infestans. So we should understand that the name RxLR type effector is derived from the presence of conserved RxLR motif in the N-terminal region of the effectors, which is very crucial for their translocation into the host plant cell. And the effectors are molecules that pathogens produce and secrete into their host to manipulate the host cellular processes and promote infection. These RxLR type effectors are secreted by oomycetes during the early stage of infection to facilitate the pathogen's entry into the host cell and to suppress the plant's immune responses. So once inside the host cell, these effectors modulate various cellular processes, alter plant defense mechanism, and create a favorable environment for the pathogen's growth as well as colonization. Next question is the favorable condition for the cause of downy mildew. There are 4 options. Out of the 4 options, the correct answer is D, that is low temperature. Besides that, we should understand what type of symptom that generally develop due to the infection of the downy mildews. Because of the infection of downy mildew, it produces the symptom like yellow spots on the leaves, fuzzy white to grey growth on leaf undersurface, then angular lesions which occur in the later stage of the disease, then leaf curling and wilting, then reduced photosynthesis, spore production especially that occurs during the humid conditions, and then impact on the fruit productions, and sometimes the infection becomes systemic in nature. Next question is on the chemical attractant from roots required for the infection by Phytophthora infestans is strigolactone. So we should understand that when Phytophthora infestans detect strigolactones in the soil, it triggers a series of events that lead to the germination of the infective spores, that is zoospores. And these zoospores then swim through the water film on the soil surface and chemotactically move toward the root exudates, including strigolactone, which act as a chemoattractant. Next question is on The trichomoniasis virus, that is potiviridae, is transmitted by, the correct answer is eraophyte mite. So other eraophyte mite transmitted viruses are, say, Escherichia kazani. It transmitted the pigeon pea sterility mosaic virus. So next question is on the aphic transmission of potato-spondyle tuber virion by encapsulation when coupled with, there are 4 options. The correct answer is potato leaf roll viruses, but not with PVX, PVY and PVS. Another question is on barcoding genetic marker used for phytoplasm identification is, the options are 16S rRNA, TEF alpha and 1623S ITS, and the correct answer is A, that is 16S rRNA. TRAI and TRAR, a regulated virulence system, is present in, the correct answer is Agrobacterium tumefaciens. Next question is, the zebra chips disease of potato is caused by Candidatus, Liberobacter, Solanacearum. The symptom produced because of this disease is very interesting, so let us see. It produces symptom on the leaf as yellowing. So zebra chips infected potato plant often exhibit yellowing or chlorosis on the leaves, which typically begins at the tips as well as margin of the leaflet, followed by leaf curling. The affected leaves may also curl upward, giving them a distinctive cupped or V-shaped appearance, followed by purple coloration is another symptom. In some cases, the undersides of the leaves may develop a purplish hue. Now let us see what type of symptom that develop on the stems. In stems, it produces purple streaking. So here, infected stem may display purple streaks or discoloration. Then followed by symptom on the tubers. In the tubers, the symptom develop is also called zebra striping. Here, the most characteristic symptom of zebra chips disease is the appearance of dark, wavy, as well as irregular striping or banding on the surface of the potato tubers. These dark stripes are often more prominent when the potatoes are fried or cooked, which makes them unsuitable for processing into the chips or fries. Then bitter taste. Potatoes affected by zebra chips diseases can have a bitter or unpleasant taste when cooked. While the external striping is the most visible symptom, but the zebra chips can also cause internal discoloration with brown streaks or flecks, sometimes present in the flesh of the tubers, and the disease significantly reduces the quality and marketability of affected potatoes. The zebra chips disease, they can lead to the reduced potato yield as infected plant may produce fewer or smaller tubers. In many cases, especially in severe conditions, these zebra chips infected potato plant may exhibit stunting, reduced vigor and delayed maturity. Most interestingly, the disease, that is zebra chips disease, is associated with the potato psyllid. The scientific name is Bacteriocera cockerelli, which act as a vector for the bacterium that causes the disease. So, monitoring for the presence of potato psyllid can be an important part of disease management. So let us see this question. Origin of yellow rust of wheat is Himalayan region of India. This is the correct answer. So as we know, This yellow rust is a very dangerous disease, and here we should understand what type of symptom that develop because of this disease. Here the Urodosori appear as bright yellow pustules chiefly on the leaves, but in severe infections they may be seen on leaf sheath too. The sori are elongated and are arranged in a linear rows between the veins of the leaf, and hence it is referred as stripe rust. Here the sori are mostly subepidermal and remain covered by epidermal layer and break only at the time of crop maturity. Interestingly, the teletosori appear late in the season and are also arranged in linear rows, and they are compact, elongated and black, which remain subepidermal. They do not break through the epidermis for a long time, remaining as a black crust. Next question is on the forecasting model used for early blight of tomato is. The options are Epidem, Fast, Tomcast, and last option is all. So here all the options are correct. So the correct answer is D, all. The chemical formula of burgundy mixture is copper sulfate plus sodium carbonate. This is the correct answer. The next question is on the reaction of effector triggered immunity, that is ETI, in plant. The correct answer is hypersensitivity response. So this is an interesting question. When PAMP in pathogens are recognized by PRRS present in plant is known as, the answer is PAMP triggered immunity or PTI. The meaning of PTI means it is a crucial aspect of the plant innate immune system, providing the first line of defense against potential pathogen. And PTI is activated when plant recognize a specific molecular pattern called pathogen associated molecular pattern that are typically conserved among different group of pathogen. PAMP are very essential compound of pathogen such as bacterial flagellin, fungal chitin or viral double-stranded RNA, which are not present in the host cell. So when a plant detects these PAMPs, it triggered a series of defense responses toward of the invading pathogen. So these responses include the production of reactive oxygen species, activation of mitosine activated protein kinase MAPKs, and synthesis of antimicrobial protein. Interestingly, the recognition of PAMs by plant receptor known as pattern recognition receptor or the PRRS initiate PTI signaling, and PRR are located on the plant cell surface and are able to detect PAMs. This interaction between the PAMs as well as PRRs activate a signal cascade that lead to the activation of the various defense gene and the reinforcement of plant cell wall to prevent pathogen penetration. So we have to understand that while PTI is effective against many pathogens, some pathogens have evolved to produce effector that suppress or evade PTI, allowing them to establish successful infection with the host. So in response to these evasions, plants have developed another layer of defense called effector triggered immunity, that is called ETI, which is a more specific and robust immune response activated when the plant detect a specific pathogen effector. So the both PTI as well as ETI play a crucial role in the plant defense, and understanding the mechanism of these immune responses is essential for developing disease resistant crop and enhancing plant protection strategies in agriculture. The next question is on which ETS is lower in expression of PTI and expression of ETI is showing. There are 4 options. Option 1 is resistance, followed by susceptibility, followed by moderate resistance, followed by moderate susceptibility, and the correct answer is moderate resistance. The next question is the dwarf burnt of wheat is caused by. The correct answer is Tilletia controversa. So Tilletia controversa produces the disease dwarf burnt of wheat, and because of the infection of this organism, it produces the symptom like smoky spore cloud, or say no external. Many a times it do not produces any type of external sign, but they produces internal infection. Then transformation of the infected kernels, followed by foul odor, followed by reduced grain quality. Then your ultimately it lead to economic impact to the crop. Next question is replication of AV sunviriodi occurs in, and the correct answer is chloroplast. The next question is on holomorph represent. The correct answer is stage that produce both meiospores as well as mitospores, that is teleomorph as well as anamorph. Some common example of holomorph fungi are Aspergillus nidulans, Penicillium chrysogenum, Fusarium graminearium, Alternaria alternata, Mycosporiella graminicola, Neurospora crassa, as well as Coletotrichum gliosporioides. Our next question is karyogamy occurs in Puccinia graminis, Forma species TTC in. The correct answer is teleospores. The next question is the Piper yellow mottle virus of black pepper is transmitted by. The correct answer is mealybug, and it is primarily transmitted by an insect vector known as black pepper thrips. The scientific name is Scritotryps dorsalis. So this is the last question of this part 2. The question is on the peanut ring spot or the bud necrosis is caused by. The correct answer is tomato spotted wilt virus. Generally, this disease is popularly known as tospovirus and are transmitted by thrips Franklinella scurgei. It produces number of symptoms, and the first symptom are visible 2 to 6 weeks after showing as a ring spot on the leaves. The newly emerging leaves are shows generally small, round or pinched inward and rugose with varying pattern of mottling as well as minute ring spots. We can see necrotic spots and irregularly shaped lesion that develops in the leaves as well as on petioles. Stems also exhibit necrotic streaks. As the plant matures, it becomes generally stunted with short internode and short auxiliary shoots. The leaflets formed on these auxiliary shoots shows a wide range of symptoms including reduction in size, distortion of the lamina, mosaic, mottling, and general closis. In advanced stages, the necrosis of the bud occurs and drastic reduction of the flowering is generally noticed, and seed produced are abnormally small, wrinkled with the dark black lesions on the testa. So thank you my friends for watching this video. Thank you.